Well, thank you for giving me this opportunity to present our work in the past few years. And the previous talks, including yesterday's talks, gives an excellent segue to what I'm going to talk about. So starting from yesterday, we talked about how AI helps science and engineering, and how does directed evolution help protein engineering to evolve a molecule. For AI's case, in general, it generates possibilities and better search space to search for new possibilities. And it relies on very rapid tests to make important progress. Similarly, in directed evolutions based on similar advantages, it generates mutations, but very rapid artificial selection evolution, and usually in single objectives. Now, what I'm going to talk about is how do we go further to redesign or remodel an organism. This is much more challenging than a single molecule or a material. Why is it difficult? Because we just don't have enough data or rules. We know insufficient about how an organism works, even though Eric Lander tried to understand everything in human beings. But we can see that even in a simple organism like bacteria, we know every single genome sequence, but we still don't know how it works. So the remaining part of my talk is going to talk about how do we improve, redesign. We don't talk about create, just redesign or remodel an organism. So in general, the way to do this is first to generate design ideas based on mechanistic information, and then you implement that in the organism. The first part may be aided by AI, it's possible. The second part, you have to implement that in the organism, and unless you have a robot, otherwise that's difficult. And then you have to rely on evolution, because no matter how you design, you get closer to what you want, but you never get exactly what you want. And many times you mess it up even more than you benefit from it. At the end of the day, you hope you discover new phenomena, new mechanisms. Hey, now, the difficulty involved in all this work is that the evolution time is long, much longer than what we like to see in today's people's attention span. Also, the gain of activity or gain of function, mutations are very rare, but these are the driving force for beneficial mutations. And we recognize that simply putting all the essential proteins together, it won't make an organism. And it has a lot of details, the affinity of the protein, the expression level, what time is expressed, the location expressed, the stability, and what's the network interact with. And you have to worry about from chemistry to reproduction to adaptation to the environment. And somebody mentioned that yesterday, the majority of species did not survive the evolution. So you can see how difficult it is. A few years ago, we engineered mice by putting in a new pathway. This is the opposite of what Dr. Reddy just mentioned. It's trying to talk about CO2 here. We're trying to burn more fat. At the end of the day, we can engineer mice to resist diet-induced obesity. Okay. Of course, this involved gene therapy, so it's much more involved. We are doing something else on that. Now we are facing a climate change problem. Okay. It's the opposite of what we just discussed. Instead of exhaling CO2, we now want to see how we can absorb more CO2 using microorganisms. And this is where we think that this type of technical solutions might eventually, not immediately, might eventually help solving climate change in a way. So if we look at the global carbon balance, the anthropogenic activities, we generate about 10 gigatons of carbon per year from fossil CO2, exactly 9.6 a few years ago. and about 1.2 gigatons of carbon, not CO2, only the carbon part, by land use change. However, most people don't recognize that we absorb 120 gigaton carbon by land photosynthesis alone. In marine, it's about similar orders, but it's much more difficult to estimate. This amount, unfortunately, half of it is going back to the atmosphere by respiration, as Dr. Reddy just mentioned. The other half slowly degrade to the atmosphere, as CO2 or carbon dioxide or methane. So this balance has been going on for a long time, and people did not know how to change that, so it's almost completely ignored. So what we wanted to do is set up a vision. How do we increase photosynthetic carbon fixation? Why do we want to do that? Immediately, we hope in the short term we can increase agricultural production to feed the increasing population, to use less land, to use less fertilizer, and so on and so forth. And the biomass we produce eventually can produce bio-oil to replace some fossil oil. And the remaining part we cannot use can be a very good carbon sink. So we begin to design the novel metabolism in plants, hoping to increase photosynthesis. We implement that, and we made some interesting discovery. On the right-hand side, we show a picture generated by ChatGPT to illustrate the landscape of evolution. So the current organism typically is on the peak of some kind of optimum, but in the local sense, not necessarily in the global optimum sense. So if we want to deal with the climate change problem, we first do a metabolic design, but most likely we mess it up. And it required further genomic changes, proteomic change, eventually developmental change to reach another peak. Another side of our vision is trying to use these ideas to do direct air capture of methane. As everybody knows, that methane is an increasingly important problem in climate change. But methane concentration is so low that native methanotrophs cannot directly use air methane. So can we engineer an organism, for example, E. coli, to capture methane from the air? So begin with the CO2 problem in plants. We know that the photosynthesis is primarily taken care of by rubisco, which is the most abundant enzyme on Earth. However, it has two activities. Through long time of evolution, it still has two activities. On the top is a carboxylation activity. It put in CO2 to fix CO2. That is called CO2 fixation. However, it cannot distinguish between CO2 and oxygen. When it put in the oxygen, it makes a glycolate, which is a non-productive product, eventually go back to CO2. This is called photorespiration. And this is the primary cause of the inefficiency of plants on Earth. Most of the C3 plants are doing this. Only a few C4 plants, including sugarcane and a few very fast-growing plants, has a special ways to avoid photorespiration. So photorespiration accounts for about 1/3 of carbon fixation inefficiency. So if you take CBB cycle, you either fix CO2 or oxygen. And 1 in 3 times, oxygen get into the way to become glycolate. And that goes to peroxisome, goes to mitochondria, eventually burn to CO2. So that is one of the major problems in the current day photosynthesis. The other problem, which is common to all organisms, and Dr. Reddy mentioned about that, that is in glycolysis, whether in plants, in human, in E. coli, or even in elephants. The way glycolysis works is first to produce a pyruvate, which is 3 carbon compound, and then produce acetyl-CoA, which is 2 carbon compound. In between, it loses a CO2. So this is one of the major CO2 sources in all organisms, and one of the main inefficiency in plants and most of the CO2 fixing organisms. So how do we deal with this? We designed a very complicated, well, for most of the non-practitioners, this could be complicated, a new cycle which we call MCG, or we jokingly call it the magic cycle. This cycle takes in the Rubisco products, two types of Rubisco product. One is glycolate, it's a C2, and convert that to acetyl-CoA without losing any CO2. In addition, it fixes one more CO2 using another carboxylase, which is PPC, which doesn't compete with Rubisco because it fixes bicarbonate, which is abundant in chloroplast. It doesn't compete with Rubisco for CO2. It also takes the 3-PG, the 3-carbon product, and does the same thing. So now we have a dual cycle system in plants to do photosynthesis and to avoid carbon dioxide fixation. So it took us a while to implement all these changes into a model plant, Arabidopsis. And the result is astonishing. You can see the wild-type plant on the right upper corner. It grew typically in this size. And we found many of the F3 homozygous plants grew 3-4 times larger that no one has seen before. If you look at it more carefully, and the biomass yield in terms of fresh weight increased by 3-fold. And this is true for 4-week-old, 6-week-old, 7-week-old plants. And if you look at the long day plants, the effect is even more pronounced. So the plants with engineered cycle grew even larger. Not only produced even larger, it has more rosette leaves, and it has produced more seeds. And all these are important factors that's beneficial to agriculture. And the roots is equally enlarged to support the biomass. So the effect is dramatic. Now we look at the photosynthetic rate, and this is the typical curve that measures photosynthetic rate. On the plateau side, that tells us the maximum carboxylation rate, it increased by 50%. And if you look at the 400 ppm level, that's the current level of CO2 in the air, it increased 100% of carboxylation rate. And why does it happen this way? Because not only it increased this metabolism, but it increased the efficiency of photochemistry. Okay, we measured this 5-PS2 activity, which for people who don't know, this is an indication of how efficient the photochemistry is taking place. And how does it increase the efficiency? Because it changed all the proteomes. It not just changed the part that we change, it changed the whole organism. At the end of the day, these are the photosynthetic apparatus. If you look at this photosystems 1, photosystems 2, electron transport chain, all the reddish ones are the increased component. So the cell responded by making more photosystems protein to support the increase of need for photosynthesis. And we can see the droplet, the oil droplet in leaves, in seeds. So these are all possible raw materials that you use to subsidize, at least substitute some of the fossil fuel, not all. And we characterize the kind of fatty acid it produces, and it's very interesting. So another story that I like to share is in the microorganism side. Here we try to engineer E. coli so that it can directly absorb methane from the air. Okay? Why do we want to do that? Because the native organisms are very difficult to do that. The concentration is too low, and we wanted to use a more model organism so we can engineer more readily. So if you compare the native organism with E. coli, we only need 4 enzymes to have E. coli to eat methane as a carbon source. And those are boxed here. If you don't start with methane, which is much more difficult, we start with methanol, okay, which is a product of methane. Then we only need 3. So more than 10 years ago, DOE started a project, invited more than 20 labs to work on this, but it was surprisingly difficult. Okay? Why it was difficult, even to grow on methanol? Because we recognize that there is a major problem caused by the intermediate formaldehyde. For people who know a little bit about chemistry, you know formaldehyde causes a very strong cross-linking reaction. So it produces formaldehyde and cross-link DNA with protein. So even if you can grow a little bit, it kills the cell at the end of the fermentation. So it becomes not evolvable. So it cannot even apply evolution to that. So we developed a way to bypass that. How do we do that? We accidentally discover a mechanism that increases the gain of activity function by a small sequence in the E. coli genome that interact with REC-BCD to promote the tandem repeats. And using that, we can tune the copy number of genes dynamically. And with that, we bypass the evolution, and after we took more than half a year to evolve an organism of E. coli that can grow on methanol. And now, this time, it grew faster than model organism. Now, the doubling time is 3.6 hours, whereas the fastest model organism growing methanol is about 4 hours. Okay? So this is already beating what native organisms give us. And we still have some way to go to make it to grow that fast in methane. We have some success, but not that fast. So eventually, we'd like to increase this so that we can use that as a bioreactor to mitigate the increase in methane concentration. And it can not, it just, it grows very nicely. Within 20 hours, it grows very high cell density. At the end of the day, we still have a lot of work to do. We don't know this MCG cycle can work in other plants or not. After all, this is just a model plant. We still need to find out whether it can work in other plants, in crops. And this synthetic E. coli, can it be robust enough to work in a bioreactor to absorb CH4, methane from the air? Finally, in the scientific side, we are pursuing the question, can design assist evolution? Okay? Instead of just evolution, can we put in a little bit of design to move the organism closer to the landscape peaks that we desire? Thank you very much.